So the last thing we're going to talk about is memory. So memory, of course, is a pretty complicated thing to understand, and a lot is still being figured out, learned about how memory actually works, which is why we're talking about it here at the end as opposed to the beginning. And in fact, we're going to kind of cover it in two chapters. So this chapter, memory system, just kind of deals with different types of memory and which parts of the brain may be involved in different types of memory. And then the last chapter deals with what's actually going on in individual neurons and at synapses where memories may be occurring. So first, let's talk about what we mean by memory itself. So first of all, there are at least two major types of memory that we can categorize and talk about. So broadly speaking, memories are either classified as declarative or non-declarative. Sometimes the words explicit and implicit are used. So declarative memories are sometimes referred to as explicit memories, and non-declarative memories are sometimes referred to as implicit memories. And then within those there are categories. So declarative or explicit memories are generally what we think of when we think of memory. So these are memories for facts and events, things you can consciously call to mind when you need to remember them. So things like what's the capital of Brazil, and then facts or memory of particular events like when what you got for your birthday or something like that. Whereas implicit memories are memories that you don't necessarily have conscious access to, but are recalled anyway. So procedural memories are a type of implicit memory, and these are memories for things like skills and habits. So if you learn how to play a musical instrument or learn how to ride a bicycle, that memory of where to put your fingers and how to make certain sounds or tones with your instrument, that is information stored in your brain. So it is a memory, but you don't necessarily explicitly remember every time you've ever ridden a bike or every time you've ever played an instrument, or every individual movement necessary to make music, whatever it is. But in any case, those memories are stored in the brain, and as we'll see, in a different way. So again, these are not just descriptive categories. They do have different correlates in the brain. And then there's other kinds of implicit memory. We'll talk about conditioning a lot more in the next chapter, but that's when a response is associated with some sort of stimulus. So that can be a behavioral response, which is when the brain or the body responds through some sort of movement due to a stimulus, or an emotional response, which is more of just an occurrence in the brain itself without necessarily the body moving. But we'll talk about classical conditioning more later on. So for now, we're mostly dealing with declarative or explicit memory. Another way to classify explicit memory is by its temporal dimension, or how long it lasts. So a long-term memory is what it sounds like. It's a memory of, again, a fact or an event that you retain for long periods of time. So that can be from months to years to your entire lifetime. So, I can remember, for example, most of the people in my fourth grade class. Now, maybe some of those memories are foggier than others. And of course, not all memory is retained permanently. So, maybe I can remember fourth grade, but I don't remember, say, my pre-K class all that well. In fact, most people don't have good memories for anything that happens to them before age 3 or 4. But for most other times in our life, we can remember those pieces of information. And then short-term memory is memory that is only good for a shorter amount of time, usually just a few seconds, sometimes up to a few hours. The difference between long-term and short-term memory, other than the time difference, is that short-term memory is vulnerable to disruption. So that means if you have some sort of piece of information and there's something that happens to disrupt it, which can be things like trauma or shock, or it can even be just some other more salient stimulus that happens from the time you learn the information. Those things can disrupt short-term memory. So for example, I can remember right now what I had for lunch today, but perhaps by tomorrow I will have forgotten, and not necessarily because I experienced trauma or shock, but because in the meantime other things, more important and interesting things will happen, and I just won't retain that memory. So there's a difference in, again, not just how long you store the information, but what kinds of things can cause it to be disrupted. And then there's another type of explicit memory called working memory. This is different from either, it's a type of short-term memory, but it's different from other types of short-term memory in that it does require rehearsal, and it has a limited capacity. So working memory is when you're really trying to store a piece of information, a small piece of information in your mind, and it requires sort of constant attention to keep it there. So the best example is trying to remember a phone number. Of course, nobody really remembers phone numbers anymore, but back when you had to do that, when you learned someone's phone number, you would repeat it to yourself over and over again, and that's the rehearsal part. And then, hopefully long enough to, say, write it down or to store it somewhere outside. But that was your working memory, and it also had a limited capacity. In fact, it's an interesting coincidence that phone numbers actually are 7 numbers, because tests on most people show that 7 numbers is about the length of a number that most people can recall from working memory. That's in fact called the digit span. So most people's digit span is about 7 digits, and anything longer than that, and it's hard to store in working memory without really extensive rehearsal and practice. But that again is just another classification. Now there's some reason to think that these types of memory are interconnected, in the sense that short-term memory can become long-term memory, and something in working memory can be converted into short-term or even long-term memory given enough practice. And there's some debate as to whether that's necessary. In other words, does a long-term memory have to go through short-term memory before it becomes long-term, or is the process of long-term and short-term memory, are those separate from each other, going through separate mechanisms? So that's just one of the many questions about memory that we don't have answers to yet. And of course, going along with memory is amnesia, which is just the loss of memory. Like I already mentioned, one type of amnesia. So again, all of us pretty much forget most of the things that happened to us in early childhood. And that's a normal type of amnesia. We all experience other types of normal amnesia. Again, if I don't remember what type of sandwich I had for lunch today, by tomorrow, that would be another type of amnesia. But usually when we talk about amnesia, we talk about some sort of pathological amnesia. So forgetting some crucial large chunk of information that otherwise we would expect to be able to remember. And so amnesia can be caused by some sort of trauma, either brain damage or degeneration, or in some cases, extreme emotional trauma can actually cause amnesia, cause loss of memory, although there's some evidence that extreme events can actually enhance certain types of memories, but generally any kind of trauma or damage to the brain will cause amnesia, and they can be classified based on where the memories are lost relative to the trauma. So retrograde amnesia is when memories are lost from a time before the trauma. So this is when, let's say you have a really bad head injury and you forget, or you have trouble remembering what was happening to you right up until you had the injury. So this is often something that happens to people who have, maybe have bad car accidents. They find themselves in the hospital, but they don't remember how they got there, because perhaps they were driving, or maybe just fell and hit their head, and they lost memory for the last few minutes or hours from before the injury. Sometimes these types of memories come back. Sometimes they don't. And often there's a gradient where the loss of memory is greatest for the time closest to the trauma, but as you go back further in time, memories become clearer and clearer. So in other words, whenever you see in the movies someone get hit in the head and then lose their memories for everything that's happened to them in their entire life. They can't remember their name. They can't remember where they're from. That doesn't really happen. So this kind of retrograde amnesia usually is just for, a few minutes, maybe a few days, maybe a few weeks from before the trauma. But usually you can still remember things that happened, say, in childhood. Also, of course, in those movies, the thing that usually cures the amnesia is another blow to the head. That is definitely not how amnesia or brain damage works. So I would not recommend trying that at home. And then anterograde amnesia is basically the opposite. It's memory loss after a trauma occurs. So, in fact, anterograde amnesia is in fact not really the loss of memory at all. It's really the loss of the ability to form new memories. So someone with anterograde amnesia usually has no problem remembering things that happened in the past. Sometimes, depending on the type of damage, they may have some retrograde amnesia. So you can have a combination of the two, but you may forget things that happened immediately before the trauma. But usually, going back further in time, you have clearer memories. But then after the trauma, you have a hard time forming new memories, being able to recall anything that happens after that. Again, sometimes this can improve over time, but sometimes it can be permanent. So again, anterograde amnesia is more about losing the ability to form new memories than losing existing memories. So the real one big question about memory is that, of course, we know that memories are stored in the brain somehow and somewhere. And so one big question about memory that has been asked for a long time is, where exactly are memories stored? So the engram is the hypothetical physical representation of a memory. So again, if a memory is stored in the brain, there must be a physical location somewhere inside your skull where that memory is actually being stored, and that's called the engram. So the question is, where is the engram for any given memory? And there have been lots of attempts to answer this question over the years. Probably the best known example is by this guy named Carl Lashley. So he did this set of experiments where he would train rats to do a maze. And it's a simple maze where they just have to get from one part of this maze to the other. So from the start to the finish. So successfully completing the maze involves starting and then going through all these turns correctly and not making any errors till you get to the finish. So an error in this case would be making one of these turns and instead of going the correct direction, going this way. So that would be an error if the rat had to stop and turn around and come back. So rats can do these kind of mazes pretty easily. After just a few trials, they learn the most efficient way to finish the maze. And so Lashley's approach was to simply say, well, if the memory of that maze and how to do it is stored somewhere in the brain, I'll just go into the brains of these rats after they're trained and start making lesions, creating damage to the brain, and just to see what happens or what part of the brain I need to damage in order to make that memory disappear. In other words, after the lesion, testing them again and seeing how many errors do they make. And these are pictures of rat cortex, so he mostly focused on the cortex. And these are different regions that he would take out. So he would go in and just take out pretty big chunks of brain. In fact, down here you can see different experiments where he destroyed different percentages of cortex. So in some cases going all the way up to 80% of the cortex would be destroyed. And indeed, the size of the area damaged would correlate with the number of errors. So the more brain damage a rat had, the more errors they would make on the test. But what he found is that no matter how many, which part of the brain he cut out, the location itself didn't seem to matter that much. In other words, he would get basically the same result no matter which part of the brain he cut out. And so the conclusion that Lashley and others have drawn from this kind of experiment is that there is really no single location in the brain where memory is stored. Now, that doesn't mean that memories aren't stored in the brain. It just means that the engram is not a singular point for any given memory. It's a distributed network of multiple neurons in multiple brain areas that is the correlate of most memory. And now, of course, I mean, there's debate about that. There are types of memory that do seem to be more localized than others, but at least for this type of memory, there was no single place where he could point to that rat would lose the memory if he took out that part of the brain. So another way of thinking of a memory, rather than being a single spot somewhere in the brain, is this idea of a cell assembly. So the cell assembly was proposed by Donald Hebb, and we encountered him before because he proposed the idea of Hebbian modification of the brain, of the developing brain, where he suggested that highly correlated activity between two different neurons increases their connectivity, and that's important for kind of fine-tuning the networks of the brain and as it develops. But this same idea can be applied to memories. So in the Hebbian model of memory, any given stimulus, like let's say you look at this circle on this screen. So when you look at that circle, that image hits your retina, and it activates a population of retinal ganglion cells that in turn activate presumably a population of neurons in visual cortex. Now, initially, that population of neurons, these neurons here, are all reciprocally connected to each other. So every neuron in this arrangement, so we have 1, 2, 3, 4, 5, 6, 7, 8 neurons, they're all connected to each other. So each one makes 7 connections to the other neurons in the group. Now, in this diagram, they happen to be arranged in a circle, but in the brain, they don't have to be arranged in a circle like this. It just so happens that the stimulus they chose and the arrangement of the diagram looks like a circle. So again, when you look at this image, these neurons are activated in visual cortex. So that's what's happening over here. You have the neurons being activated when that stimulus is present. And as long as you are looking at that stimulus, that same population of neurons is going to be activated. And because they're all connected to each other, so they weren't activated by each other, they were activated by inputs from the eye. But because they're connected to each other, they create this reverberating activity, which means that as they're all active, they all send signals back and forth to each other in this kind of reverberating pattern. So all of them communicating with each other over and over again. So the signal is just bouncing back and forth within this network. In Hebb's model, this reverberating activity is basically what's happening when the brain is storing a short-term memory. So storing a short-term memory means somewhere in the brain there is a circuit of cells that's in this sort of constant state of activity, and that reverberating pattern is the memory, essentially, that circuit being turned on. And if that activity, that reverberating activity, is active long enough, eventually the connections between those neurons will get stronger. This is again Hebbian modification. So whenever you have correlated activity, so as long as all these neurons together are active at the same time, their activity will be correlated, and so their connections will become stronger. So here they just drew the lines thicker, but these represent stronger synapses. So through LTP or through some other mechanism, these neurons become more and more well connected to each other, and perhaps less connected to other neurons that are not as well, whose input or activity doesn't correlate with them. Now, what's important now is that as the synapses become stronger, that creates a effectively permanent change in those neurons. It's not just an increase in activity. Now, perhaps the dendrites form more connections with the axons. Maybe there are more receptors being expressed on the synapse. Maybe the axons are releasing more neurotransmitter. But the point is there's a permanent change in these neurons that allow those connections to remain strong even when the stimulus goes away. So now down here, the stimulus is gone, so the activity in these neurons goes away, but the increased connections are still there. So again, in the Hebbian model, this would be the long-term memory. So the long-term memory is just the increased synaptic strength between these neurons that make up this circuit. And then recall, meaning actually being able to remember a stimulus, would be either full or partial stimulation of that same circuit, which is either triggered by the original stimulus or maybe just some subset of a stimulus. So perhaps instead of seeing the entire circle, you just see 4 little arcs. And that doesn't activate the whole circuit right away. It only activates a subset. But because the connections between these neurons are all strengthened, activating that subset will eventually activate the whole circle. And that then allows the original stimulus to be recalled or brought to mind. So again, this is a very simplistic model for how these circuits might work. Obviously, you would need a lot more neurons and a lot more connections for more complex memories. But this is just a way of kind of introducing the idea that memories are again not stored in individual locations, but are stored in circuits of neurons that may be spread out. So again, in this picture, the cells are all in a circle right next to each other, but in reality, in the brain, these neurons might be spread over a large part of say visual cortex, or you could even have neurons in different completely unrelated parts of the brain, parts of cortex that are involved in storing that memory. Now, that doesn't mean that there aren't parts of the brain that are more important for memory than others. There are parts of the brain that are more important, if not for storing memory, at least for forming new memories. In fact, if you go back, it's not surprising when you look at the parts of the brain that Lashley cut out. So, especially these posterior parts of the cortex, that's where a lot of the sensory regions are. So you would not be surprised to find that memories for sensory information would be stored in the sensory cortex. So again, there's no single region that is responsible for storing memories, but there do seem to be parts of the brain that at least are important for forming memories. The temporal lobes, especially the inferior temporal lobes, are one of those regions. So we've seen the inferior temporal lobe before. It is connected to visual cortex. We know that damage to this cortex specifically damages or impairs the performance of monkeys on visual memory tasks, where they're given an image and they're expected to recall it later. Also, we know that there are face cells in this part of the brain, and that not only do they recognize faces, but they can sort of learn faces. So if you say are recording from an inferior temporal lobe neuron, like they're doing here, and then showing an image of a face to the monkey over and over again, some neurons will sort of learn particular faces. So in this case, they've shown 4 different faces to one part of the brain, and one particular face seems to show increased responsiveness to in that neuron over time, which implies that there is a memory associated with that face that involves at least the neuron that they're recording from. And so that's one of the ways you kind of measure memory in animals is just looking to see, does the animal or does the neuron in this part of the brain have a different response to repeated presentation of a given stimulus. Another thing about the temporal lobes is that if you damage the temporal lobes, especially the medial aspect, the innermost part of the temporal lobes, you get declarative anterograde amnesia, which means people with this type of damage lose the ability to form new memories. The famous example of this that we talked about way back in the beginning of the class is HM. So HM had, had epilepsy, and to treat it, they removed the medial temporal lobes on both sides of his brain, and he lost the ability after that to form new memories. Every day he had to be reminded of what had happened to him the day before. In fact, he couldn't really remember anything that happened after he had the surgery, mostly for the rest of his life. But he only really lost the ability to form declarative or explicit memories. Non-declarative memories he still could form and retain. So he could learn new tasks, like they would give him various procedural memory tasks to complete and practice on. Now, like anyone else, he had to practice the task over and over again to get better at it, but he could do it. The difference was he couldn't remember having learned it in the past. So he would have a task, he would be told to do it. He would say, "I've never done this before," but then he would do it as well as someone who had been practicing, for days or weeks, but would have no ability to explain how he had learned that. So again, it's what that means is that the bilateral temporal lobes, medial temporal lobes, are important for at least formation of declarative memories, but not for formation of non-declarative memories. And again, it did not wipe out the memory itself. So his memories for things that happened before the surgery, so going all the way back to childhood, were perfectly intact. So the memories aren't stored in the medial temporal lobes, but somehow these regions are important for forming new memories. So what's in the temporal lobes, the medial temporal lobes? There are three major structural features of the temporal lobes that are important. One is this thing called the hippocampus. The word hippocampus comes from, I think, the Latin word for seahorse, presumably because in cross-section it kind of has this horse-looking shape to it. But basically it's just a folding of the cortex, the temporal lobe, that goes all the way back and kind of wraps around. So it's in the medial temporal lobe, and if you kind of look at it from the side, you can see it goes all the way back and kind of curls up to the very end of the temporal lobe. And then there's some surrounding cortical structures. So right next to the hippocampus, you have this region. So here's the hippocampus here. And then right next to that, you have what's called the entorhinal cortex. And then next to that, the perirhinal cortex. These are referenced by the fact that they're right next to this groove here called the rhinal sulcus. So the rhinal sulcus is right here, and it's just a sulcus on the medial surface of the temporal lobe, and so the cortex above it is called entorhinal. The cortex below it is called perirhinal. And then wrapping around the rest of the way, you have a region called the parahippocampal cortex. So all three of these structures are important for memory formation. They all get extensive input from the cerebral cortex. So the cortical association areas, so remember that's the parts of the cortex that synthesize sensory input from different regions, and that sends information to these medial temporal structures, cortical areas, which in turn send information to the hippocampus, and then there's a large projection from the hippocampus back to the cortex, and then through this white matter bundle called the fornix to the thalamus and hypothalamus, which is also important for processing sensory information. So this is just the structure of the medial temporal lobes. So, of course, from HM, we know that if you damage these regions, you lose the ability to form new memories. In animals, you can show with a little more precision what happens. So in monkeys, monkeys have a hard time with this task after hippocampal or after temporal lobe damage with something called the delayed non-match to sample. And so this is a pretty simple task where it's abbreviated DNMS, but in the delayed non-matched to sample task, first a monkey is trained to move an object to get a reward. So it's a simple, the cage of the monkey is put in front of this board with 3 little wells, and then you have an object that covers the well, and it can just be like a little block or piece of wood or whatever, but it has to have a distinctive, some distinctive feature to it. So it could be different colors, different shapes, doesn't really matter. And the monkey just has to move it out of the way to get, a piece of candy or a piece of fruit or whatever. So monkeys will do this pretty easily, or can be trained to do this pretty easily. But then the task is, after they've learned that they're supposed to move the object to get the treat, then you lower a screen, so you block the monkey's view or maybe move the board out of the way, either one. And then you wait. So there's a delay of some amount of time, and you vary the amount of time. But then while the monkey can't see, you put the same object from before. So, if you put the red circle out, then you put the red circle back. But this time, instead of in the middle of the board, it's on one of the ends, and it doesn't matter if it's right or left. And then at the other end, you put some new object, something that was not present in this phase. And so we call this object, the one that the monkey sees before the delay, the sample. And the task, and what you've done also during the delay, is you put a treat in the well under the new object. So this is the object that's not matched to the sample. So that's the idea, is that the monkey has to remember which object it saw, and it has to choose the new object. So it has to, that's why it's called a non-matched to sample. And monkeys with medial temporal lobe damage can do this. They can be trained on this task. But the delay causes a big impairment. So this graph shows the success of normal monkeys at different times of delay, and then monkeys with temporal lobe damage. So for normal monkeys and monkeys with lesions, if there's a very short delay of less than 10 seconds, both monkeys with and without damage basically do the same. They get about 90% correct, and monkeys with no damage can still get 90% correct even after a delay all the way up to 10 minutes. So sitting there just waiting behind a screen for 10 minutes, normal monkeys have the ability to remember later what they saw before the screen went down, or which object was the object that they saw before. Whereas monkeys with temporal lobe lesions start to do very, very poorly once there's any amount of delay. So even at 15 seconds, a minute, and after 2 minutes, just forget about it. At that point, their percentage correct is getting close to 50%, which of course is just chance. So if they have no memory at all, if they just randomly pick an object, they'll get it right about 50% of the time. So 50% means basically they have no memory for what they were seeing before. So this is, again, just evidence that the temporal lobes in this case are important for storing this particular type of memory, just remembering what type of object they saw before the delay. And of course, this is short-term memory; it's only a few minutes, but there are other studies that show that long-term memory is also impaired by this kind of task. So the temporal lobes, again, they're connected to the cortex. They're also connected to other parts of the brain, which seem to be important in memory. The fornix, again, is the white matter pathway that comes out of the hippocampus, and it sends a projection to this little structure down here called the mammillary body, which is on the ventral surface of the diencephalon, just right behind the hypothalamus. But then there's the mammillary body in turn projects to the thalamus itself. And it turns out that damage to these parts of the brain also cause memory impairment. There's a condition called Korsakoff syndrome that's usually caused by excessive alcoholism, and among other things, this causes damage to, actually the alcoholism leads to poor nutrition, which causes a vitamin deficiency, and the vitamin deficiency causes brain damage. And in some people, it causes brain damage here to the thalamus and other parts of the diencephalon. And people with that kind of syndrome have a lot of memory, declarative memory impairment. So again, the projection from the temporal lobe to the thalamus seems to be important for the ability of the temporal lobe to form memories. So again, all the different parts of the medial temporal lobe are important. The hippocampus is probably one of the most important parts and gets perhaps the most attention. So we know in the hippocampus that neurons in this part of the brain in humans are particularly sensitive to familiar stimuli. So if you record from neurons in the hippocampus and present the patient with any kind of sensory stimulus, these neurons don't necessarily respond. So they're not sensory neurons. They don't respond to sensory stimuli unless it is a stimulus that the patient has seen before. So a repeated stimulus is going to activate a lot of hippocampal neurons more than new stimuli. So again, that's an implication that the memory of the image or the object, whatever the stimulus was, at least in part involves the hippocampus. In rats, damage to the hippocampus causes specific kinds of memory impairment. So this task here requires hippocampus. It's called the radial arm maze, and it's called that because it's a pretty simple maze. It's really just kind of a cylindrical box here with 8 arms coming out, and at the end of each arm there's a little piece of food. So the experimenter just puts the rat or the mouse into the center and then lets the rat explore, and most rats will immediately start going down all the arms and start retrieving the food. And so again, both rats with hippocampal damage and those without have no problem going down, getting the food, and exploring the whole maze. But to test the test here involves seeing if and even if you only put food down some parts of some arms, both rats with and without brain damage will only go down the arms that have food. But rats that don't have brain damage will only explore each arm once. So they will go down one arm, get the food, and then go to all the other arms. Then when they're done, they just wait in the center. Whereas rats with hippocampal damage will go down each arm, get the food, and then they'll have a tendency to go back down the same arm again. So they can't seem to remember that they already went down or which arm they already explored. And so they end up kind of visiting the same arms over and over. Another type of memory task that involves the hippocampus is this water maze. So the water maze is also pretty simple. It's just a big pool of water, like a little kiddie pool, and somewhere in that pool there is a platform that is just below the surface. And so if you're a rat or a mouse swimming in this water, you can't really see the platform. You can only see it if you're standing above it. And rats and mice, of course, can swim, but they don't really like water. So they will immediately try to find the platform. They'll just swim around until they find it. And after only really a couple trials, most rats will eventually find the platform and remember where it is and go to it immediately once they're put in the pool. But rats with hippocampal damage have a hard time remembering from trial to trial where the platform is. So it just takes them longer to get there. So it's just another way of again, kind of measuring explicit memory. Also, in the hippocampus, there are cells that seem to be active whenever the rat is in a certain place. So that means that there's basically place cells. So here, what they're doing is they've got the rat in this special cage that has partitions, and the rat's basically able to freely explore. But what they can show is that certain neurons in hippocampus will become active whenever the rat is in a certain location. So cell 1, for example, seems to be active whenever the rat is in this region here that's just above the partition, whereas a different neuron is only really active when the rat is in this location on this part of this partition. So perhaps the hippocampus, or at least at the very least, the hippocampus is important for spatial memory, the ability to remember where things are, where one finds oneself in space. And so that's just one type of memory, but it definitely seems to involve the hippocampus. Other parts of the brain that are involved in memory. So again, mostly we've been talking about types of what we would think of as explicit or declarative memories up here. But procedural memories also involve other parts of the brain. So the striatum seems to be involved in procedural memories. We talked about the striatum before. Of course, its main job is usually we think of as helping facilitate movement. So obviously damaging it impairs movement, but damaging it also impairs certain types of memory, especially, and so this task shows that. So damaging the striatum does not impair performance on the standard radial arm maze. So rats with damage to the striatum, this is human striatum, but the rat striatum, if it's damaged, does not impair performance on the standard radial arm maze, which means they can still remember which arms have food and only go down them once. But there's a variant called the lighted radial arm maze that rats with striatal damage have more trouble with. In this one, again, the food is placed at the end of these arms, but not all of them. Only certain arms have food, and those arms with food have some sort of visual cue. It might be a little light that comes on, or it could be something else. But there's some sort of extra information that tells the animal which arms have food and which ones don't. And from trial to trial, the lights move. In other words, which arms have food vary and are random from one trial to the next. So in other words, the rat can't memorize which arms have food and which ones don't. What they have to do is memorize the task. Only go down arms that are lit up. Don't go down arms that aren't. So again, normal rats can figure this out pretty fast and will not waste time exploring the arms that don't have lights on. Whereas rats with striatal damage can't seem to remember what they're supposed to do. They know there's food on some of the arms and that they need to figure out which ones they are, but they can't remember the rule that you only need to go down the ones that have lights on. So again, that's implied to be a type of procedural memory. Because it's again memory of a rule, not so much memory of a particular location. And of course, the example in humans of striatal function is Parkinson's disease. So Parkinson's is not damage to the striatum, but damage to the substantia nigra, which has kind of an excitatory input, excitatory modulation of striatum. So people with Parkinson's, in addition to motor impairments, also do have impairments of procedural memory. So this is a task where to test procedural memory in people, where you're given a set of cards, and they're just these patterns that show up on the cards, and they're supposed to be cues for you to predict the weather. And I'm using air quotes that you can't see, but they're just asked, okay, based on these cards, predict the weather. Now, of course, they have no idea what the cards mean, and the scientist, the experimenter, doesn't tell them what they mean or which cards correspond to which weather patterns. But they're told after they choose whether they were right or wrong. And so most people, after doing this for many, many trials, will eventually sort of figure out, again, implicitly, not necessarily explicitly, which cards represent or predict which types of weather, and they get better at it. Even people with other kinds of amnesia, in other words, people with like someone like HM or someone else with other kinds of explicit amnesia, will be able to get better at this task. That's what this graph over here shows. But people with Parkinson's basically don't ever improve. But people with Parkinson's don't seem to have any problem with explicit memory. So if you give them an explicit memory task, like just to remember which image they saw or which card they saw, they do about as well as normal patients. Now, working memory, meanwhile, is also complicated and also seems to involve lots of different parts of the brain. But the prefrontal cortex seems to be especially important for working memory. So first of all, the prefrontal cortex gets a lot of input from the medial temporal lobes through the thalamus. So there's a connection there. And we know that, again, damage to the prefrontal cortex causes certain types of tasks to be impaired. So in this case, this is called just the delay task. So it's a simplified version of the delayed non-match to sample. Here, the monkey just has to remember from one moment to the next what image they saw. So again, they're shown one object that is hiding a treat, and then there's a delay where the screen's brought down, and all they have to do is wait until the screen comes back up, and then move the object to get the treat. And again, monkeys so the idea is that while the screen is down, they're having to remember which well had the treat and which one didn't. And so monkeys with prefrontal cortex damage have an impairment with this type of task. In humans, a way to test working memory is this task called a Wisconsin card sort. And there's other ways to measure working memory, but this is just one. Again, the subject just is given some cards with these different shapes and colors and number of and patterns on them, and they have to sort them either by color or by shape or by number. But again, they're not told which is the correct way to do it. And of course, the correct is just whichever one the experimenter decides is correct. And so they're expected to do this over and over again until they get it right. Just keep sorting until they get their correct pattern. But then the rules change. So once after a certain number of trials, which pattern is considered correct changes. Again, the patient isn't told that. And then they have to learn a new sorting rule. So normal people, again, after a few trials, eventually get better and better at this, but people with prefrontal cortex have a hard time. So they can learn that there is a pattern, but they have a hard time changing to the new rule later on. So it's kind of dealing with new information and keeping that information in working memory that they seem to have trouble with. Again, the probably famous example of someone with prefrontal cortex damage that we talked about was Phineas Gage. He was the guy that had the tamping rod go through the front part of his brain, and so working memory and these kinds of rule changing tasks is hard for people with prefrontal cortex damage as well. There is evidence that during some kinds of tasks where there's a delay, neurons in prefrontal cortex can be active. So here they're just recording from a prefrontal cortex neuron during some sort of delayed task, and there's a cue period where they're shown the monkey or the patient is shown whatever it is they're supposed to remember. There's a delay, and then a choice period where they have to go back and remember what they were seeing in the cue, and select it. So some neurons in prefrontal cortex are active during that delay. So that means that during the delay period, they're in fact more active than they were during the cue or the choice period. The implication is that this neuron is part of the circuit that perhaps is storing the cue information. But not all neurons in prefrontal cortex behave this way. So, presumably only those ones that are part of that particular memory. Again, and this is reinforced by the idea that there's not any one part of prefrontal cortex that is necessary for any one type of memory. Different parts of prefrontal cortex seem to store different types of memory and different types of information as working memory. So there are at least 3 areas in prefrontal cortex that are involved in facial working memory or memory of faces, and then a couple different regions that are more important seemingly for working memory for spatial information. So again, there's not any one single spot that is important for one type of memory. And then aside from prefrontal cortex, or there are other cortical areas that seem to be involved in working memory that are connected to prefrontal cortex. One we've talked about before, so the premotor area, which is part of the motor cortex. For example, in this experiment, remember the monkey's task is to see this instructional stimulus, wait until it comes on, and then hit the corresponding button once the trigger stimulus comes on. And there are neurons in the premotor area that are activated when the instruction stimulus comes on, and stay active during this delay from when the instruction stimulus comes on to when the trigger stimulus comes on, and then they sort of go quiet once the trigger stimulus is activated. So the implication is that the memory, both the memory of which button to push and, since this is motor cortex, how to actually engage in the movement that's about to be required, is being stored in this circuit. So this may be part of the working memory circuit for this particular task. And then there's another area called the lateral intraparietal sulcus. So this is a region in the parietal lobe that also seems to be involved in storing information in a short term period. So here is a pretty simple memory task where it can be done again with monkeys or humans, but in this case the monkey is shown a screen, and the task is to fixate on a point in the center. Usually there's like an X or a little crosshair in the center, and again the monkey can be trained to do this just as long as there's a camera that's watching it, the eyes, and there's software that can track where they're looking, and as long as they're looking at the right place, then they get a reward of juice from a dispenser or whatever. And the task is a target image is flashed somewhere on the screen. So it's just some random place on the screen. It can be just any little image, like a little square in this case. And then it goes away, and the monkey is trained to wait until after the object goes away, a certain delay time, and then to look at that position. So it's it's a spatial memory task, because you have to remember the location in space where the image was, even after it's been taken away. And so again, this is an example of working memory, and this task seems to involve this intraparietal sulcus area. So neurons in this region will be active during this delay. Anyway, that's, again, just a real brief kind of introduction overview of different types of memory, different parts of the brain involved in memory. In the next chapter, we'll get into a little more detail about what's actually going on at the cellular level.